question after the talks. And I want to let you know that all the hosts here are also trainees in School of Biological Sciences. Okay. So without further ado, I'll just pass it to our first host, Maggie.
Hello, everyone. I am pleased to introduce my lab mate and mentor, Meixue, Duan. She's in her final year of her Ph.D. in Dr. Gregory Gibson's lab, and she's originally from Chengdu, China. She came to Georgia Tech originally as a master's in bioinformatics, and she loved it here so much that she wanted to pursue her Ph.D. She works with immunologist Dr. Anyang Lee over at Emory, along with Dr. Ignacio Sanz. And she works on deciphering short and long-lived plasma cell survival for the basis of effective immunity using single-cell transcriptomic analysis, and she'll be sharing that today. So please give me a warm welcome and introducing Michelle.
Thank you, Maggie.
Hi, everyone. So I'm Meixue Duan. Yeah, I'm from Greg Gibson's lab. Today, I'm gonna share some of the results from that single-cell study, which shows the distinct TNF signaling pathways in human bone marrow long-lived plasma cell maturation. Today, first of all, I will give a brief view of today's talk. First of all, I will give a short introduction to what are long-lived plasma cells, then how heterogeneous are human bone marrow plasma cells, and what pathways and genes distinguish different maturation paths, how survival genes are expressed in those bone marrow plasma cell subgroups, and finally, a very short summary. First of all, I will give a short introduction to what are long-lived plasma cells. So from Only's lab in 2015, they published a paper and found that the long-lived plasma cells are contained with CD19 negative, CD38 high, and CD138 positive cell populations. They use the cell surface protein CD19, CD38, CD138 to subgroup the antibody secreting cells, and they use the number 235 and the character ABD to distinguish those subgroups from the peripheral blood and the bone marrows. Sorry? Okay. Yeah, it has to be there. Yeah, yeah. Can I close it first and then open it after the presentation? Maybe here. Yeah, here we go. All right. So in that study, they did some tetanus-specific antigen detection, measles and mumps-specific antigen detection, as well as the flu-specific antigen detection. So all those subjects recruited for this study, they received the flu vaccine 1 to 11 months before they give the bone marrow aspirates. And you can see that for those subjects which, can you see? Yeah. Which recruited to shows the higher, like the serum titers for the tetanus and the measles mumps specific antibodies. Actually, they didn't receive the vaccine. They don't have any history of the MR vaccine, but all those antibodies are from their childhood infections. So you can see that even after tens of years, within the Ph.D. cell populations, they still can detect some specific antibodies. So from this study, the summary is like the non-lived plasma cell can exist for a lifetime, which contained in Ph.D. cells, and secrete high affinity antibodies that maintain steady state Ig secretion in the absence of the antigen. Then I'm gonna talk about how heterogeneous are human bone marrow plasma cells. To answer this question, we did the single cell study, and all these, like the library preparation, cell fact sorting stuff are done by CDSCNE. And first of all, we recruit we get 5 healthy bone marrow aspirate. And we use the previous defined the cell surface markers to sort our population our targeted cell populations, POP-A, POP-B, and POP-D. And we use the 10x genomics to build the libraries for the single-cell RNA and the single-cell VDJ libraries and send out for the sequencing. After the very carefully QC step and the test for the cluster stability, finally we discovered the 15 subgroups of the bone marrow plasma cells. All those 15 clusters we detected in all 5 subjects. And for those clusters, we always can detect some markers to represent the cell population. And here, I just want to mention that for the cluster 1, which has the highest expression of the MK67 and the CD43, which is encoded by the SPN, these genes. And the marker gene expression is consistent with previous defined the plasmablast cell population. And except for the cluster 4, we only detect some immunoglobulin genes highly expressed in this cell population. No other informative genes highly expressed. So we think the cluster 4 should be like the intermediate stage of the plasma cell maturation. And here after we will call the cell cluster from 1 to 4 as the early stage of the plasma cell maturation, while 5, 6, 7, 8 as the late plasma cell maturation. The cluster 9 and 10, because they shows the interference gene signatures, and we call them as the interferon responsive cell population. Cluster 11 shows the higher percentage of the mitochondria encoded mitochondria transcript, and we call it the mitohigh cell population. Cluster 13, 14, and 15, which has a higher proportion of the IgM isotype cell population, and we call them the IgM population, which I will show more details in the following slides. First of all, we show some of the representative gene expression in the current cell clusters. MK67 highest expressed in the plasmablast cell population. Then the CD19 gene expression we see is mostly expressed at the bottom of this AAC island, while CD138 genes shows the highest gene expression in the top of this island. These two known like the transcription factors, which are very important for the plasma cell maturation. We see the XBP1 shows a very stable gene expression during the whole maturation process, while the BNIMP1, which is encoded by the PRDM1, will show the higher expression in the early phase and a slightly higher gene expression in this part, while decreased gene expression in this group of cells. And we count the proportion of the FACS-sorted cell labels in the current cell clusters. We see the early phase of the plasma cells has a higher proportion of the POP-A and POP-B cell population, while late phase of the plasma cell is mostly combined from the POP-B and POP-D cell population. Similar trend we see for the IgM dominant cell population. What's interesting is we still see there are some small proportion of the IgA cell population located in the late phase of the plasma cell. Actually, when we show the gene expression, we see this group of the POP-A actually transcriptionally they resemble POP-B cells as CD138 encoded. No, the SDC1, which encoded the CD138, shows the intermediate gene expression. So the current assumption from our lab is like, if the plasma cell wants to become as the non-living plasma cell, first of all, they need to exit the cell cycle process and start maturation like to the non-living plasma cell, as well as during the process, they should diminish the B cell phenotypes. So here we show the gene expression for the DBTB32, the inhibitor of the MHC class 2 gene, and the LYN, the inhibitor of the BCR gene. So we see these inhibitor genes highly expressed in the early phase, as well as the MHC class 2 highly expressed in the early phase of the plasma cell maturation. While there is an exception, which is HLA-DOB, which increased the gene expression during the plasma cell maturation. Actually, the previous publication shows that this gene is not regulated by the C2TA, which is the main transcription factor that regulates the gene expression of the MHC class 2. So this key map is from the bulk RNA-seq data, which is generated from the in vitro cell culture, and all this data is generated by Aiello from An's lab. And she used some blood RNA-mediated ASC put into our cell culture medium. And from each time point, you can see like the same time point. So she saw some more cells and do the bulk RNA sequencing. You can see the gene expression actually, I think, match quite well with the single cell observed expression pattern. This is the summary from the VDJ analysis, and we count the fraction of the isotypes in current cell clusters. So we found that the majority of the bone marrow plasma cells are IgG1 dominant cell populations, while cluster 13 to 15, we see there has a higher proportion of the plasma cells contain the IgM isotype. That's why we call it the IgM dominant cell populations. And we use other genes like the CD19, B220 to help us characterize the progenitor of those two dominant lineages. And in the previous publication, it shows that the B220 has a higher expression in the newly formed bone marrow plasma cells. That's why we see we like label like the cluster 1, 2, 3, 4 as the early phase of the plasma cell maturation, as well as the cluster 13. We think it should be like the progenitor cells for the IgM's dominant cell population. And the J chain, as expected, has higher expression in the IgM dominant cell populations. So the current well-known accepted like assumption for the non-living plasma cell is the plasma cell generated from the germinal center, they are more likely migrate into the bone marrow and become as the non-living plasma cells. So the germinal center derived plasma cells, they are class switched, they have higher hypermutations, somatic hypermutations. That's why we checked if we can, by characterizing the mutation frequency in the current cell clusters to help us distinguish like the early phase of the plasma cells versus late phase of the plasma cells. So the NB cell population is the naive B cell population, which we used as the control to compare with the plasma cells. So you can see naive B cells compared with plasma cells has a really low mutation frequency. And the cluster 6, no matter when we count the global V region mutation frequency or by the CDR region, by FR region, we always see cluster 6 has the average higher mutation frequency than any other cell populations. And we did the permutation test to validate actually cluster 6 has significantly higher mutation frequencies. So this is one characteristic we need to keep in mind. And the other, so in each cell cluster, we also calculate the mutation frequency by the isotypes IgG, IgA, and IgM. So for the IgG and IgA, we see very similar mutation frequency, but they are significantly higher than the IgM cell populations. Next, I'm gonna talk about what pathways and genes distinguish different maturation paths. So by the trajectory analysis, we predicted 4 paths of the maturations. Path 1 is from the cluster per PB stage to the cluster 6 highest mutation frequency. Path 2 is to cluster 7 and 8. Path 3 is to the interferon positive. Path 2 is to the mitochondrial population. And we want to know what kind of pathways can distinguish those 4 paths, those 4 maturation paths. So we did the enrichment score using the current 15 hallmark pathways from the GSEA database. And we projected the enrichment score on the current UMAP, so you give you a more intuitive way, like on like how the pathway enrichment score changes during the maturation. This fitting nice shows the trend of those enrichment score in 4 paths. So what I want to highlight here is, so you can see most of the pathways decrease the enrichment score from the early to late phase maturation, while we see the TNF-alpha signaling NF-kappa B pathways shows a really increase in our path 2, while keep like the very stable enrichment in other 3 paths. So we want to know So first of all, we want to know if what we observed the enrichment is by any like the donor biased expression or the outlier gene expression. So we checked like the enrichment score in all 5 individuals. Then we see the enrichment score decrease in the cluster 6 while increase in the cluster 7. And the gene expression shows the general like the decreased gene expression in the cluster 6 while increase the gene expression in the cluster 7. So the TNF-alpha signaling by NF-kappa B this pathway really help us distinguish those 2 paths which might contain the long-lived plasma cells. And to further characterize like those 2 paths, what kind of genes can help us distinguish them. So we see there are 80 genes that specifically upregulated in the path 2 cluster 7, and which include the Duan, B, phosphorus B, MCO1, BCO2. NF-kappa B, IA for those genes, while in the cluster 6, which is in path 1, we see the CD138, CD9, CD63 highly expressed in the path 1 cell clusters. And this is from the 2021's review paper. So it shows that the NF-kappa B, IA, TNF, AIP3, these genes are highly expressed when the NF-kappa B pathway being activated. And also, NF-kappa B pathway activation is not just solely being activated, usually combined with some other pathways like the Duan-K, JKP38, which can also upregulate the gene expression of the Duan, FOS, and the Duan-D, those genes. So next, I will show the survival gene expression in the current plasma cell subgroups. So I will give you, this review paper shows that, generally like the MCO1, BCO2 is the pro-survival genes, but the BEAM is the activator of the apoptosis. If the pro-apoptotic genes has higher expression than the post-survival genes, this cell population is more likely will prime for apoptosis. But there is another cell population called the apoptosis refractory cell population, which doesn't have the pore-forming genes, which is the BAC and BAX and BAC1. So this cell population, no matter like how high the pro-apoptotic gene expressed, they are not likely to primed for apoptosis. So here is the genes from the BCL2 family, and I group them as the pro-survival, pro-apoptotic, extrinsic pro-apoptotic, and the cell cycle arrest gene expression. So what I want to highlight here is for the MCO1, BCL2, you see the gene expression decrease in the path 1, cluster 6, while increase in the path 2, cluster 6 and 7. But for the cluster 12, which really capture eyes, has the highest expression of the pro-survival genes as well as the highest gene expression of the intrinsic pro-apoptotic genes, and we think this cell population is very stressful plasma cells and might prime for apoptosis. And this is some like the gene expression trend in the current 4 path. So the MCO1 really distinguish like the path 1 from the path 2. So in the path 1, the MCO1 gene expression decreased in the late phase while it keeps going up in the path 2. So the plasma cells, as I showed before, so although the path 1 decreased the MCO1 CD1 BCL2, but it has the highest expression of the CD138. So we are wondering if the non-native plasma cell can use some other, like the pro-survival genes, to help them to be non-native plasma cells. So these figures shows from the VDJ data analysis. Just basically want to show you, yeah, we have some from the VDJ analysis. We do observe some clones, mainly located in the early phase, and some start from the transition and to the late phase, and some in the late phase. And there is one clone started at the very beginning and matured to the late phase to support our inferred trajectories of the plasma cell. And from this circus plot, you can see that each clone is colored by the most matured cell populations. So you can see within the early phase, you will have higher clones that colored by the early phase cell clusters, while there are still a small fraction of cell population or clones, they can further mature into the late phase. Mercida index also shows that the cluster 1, 2, 3 early phase, we see the higher connectivity and the similarity. Late phase 5, 6, 7 also shows a higher connectivity and similarity. So in summary, as the human bone marrow cells are very heterogeneous and can be represented by different of the cell markers. And the human bone marrow plasma cells, they are IgG1 dominant as well as the IgM dominant cell population. So human bone marrow plasma cells may have different progenitor cells parity refill the plasma cell pool, as I showed in from the VDJ results and the gene expression. And we inferred several maturation paths for the human bone marrow plasma cell maturation. And the non-native plasma cells also very heterogeneous and can use maybe different survival factors to promote its survival. Yeah, thank you. Thank you for my collaborators.
I know we're over time a little bit, but if anybody has questions for Michelle? Anybody? Has the chat? The messages on the chat? Okay.
Thank you. Yeah, I will pass the.
Okay, we'll pass it on.
Yeah. Thank you very much.
All right, can everyone hear me? All right, it's my pleasure to introduce Zach today. He's a postdoctoral fellow in the Streelman lab here at Georgia Tech. He earned his Ph.D. at Emory University, where he studied the neuroscience of constellation behaviors in voles and other rodents. And here at Georgia Tech, he continues to study behavioral neuroscience using cutting-edge tools and techniques. Please join me in giving a warm welcome to Zach.
All right. Thank you, George, and thanks everyone for coming, and thanks Dr. Ham for organizing this. I'll get right into it. I am really interested in how natural genetic variation and the brain coordinate the incredible diversity of social behavior that we see in nature, and also variation in social cognition and behavior that exists across humans. But one of the major challenges for behavioral neuroscience ever since its inception is the incredible complexity of the brain and also the incredible complexity of the genome, but I'm going to focus on the brain today. And one of the major hurdles that has always faced behavioral neuroscience is sifting through this haystack to find the important needles. And that's really difficult, and what the whole field has kind of adjusted around that challenge and has converged on model organisms and pretty expensive, laborious experiments that require a lot of time and usually teach us a lot about only one cell population or molecular signaling system at a time. So I'm going to talk about how I'm tackling this challenge in Lake Malawi cichlids. You may be familiar with these fishes. They're a really amazing and powerful comparative model for understanding the genetic basis of complex traits. They've explosively radiated into thousands of species over a relatively short evolutionary time span. They're phenotypically diverse, but I'm really interested in their behavioral diversity, and they're genetically not much more different. Species are not much more different than individual humans on average. And because of that genetic similarity, they can be hybridized, and really powerful experimental genetic mapping approaches can be applied. Today I'm going to talk about a behavior that's called castle building, which is a really interesting behavior in Lake Malawi, where males during the breeding season will construct large underwater courtship structures out of sand, and they spend a lot of time and energy doing this. And the lab has previously traced species variation in bower construction behaviors like castle building to a region of the genome. So castle building has a genetic basis. And we can try to understand how genes in the brain are coordinating this behavior. But in order to study this behavior systematically in the lab, you have to have a system for measuring it. And I spent the first few years of my postdoc doing this. It's not trivial to establish a new behavioral neuroscience system in the laboratory, a new species and a new behavior. So I won't get into it, but our system works with a Raspberry Pi, a depth sensor that tracks the structure, and action recognition, a machine learning approach for video data that's able to pick up many different building, quivering, and other feeding behaviors. And so we run this system 24/7, and we're able to get a lot of information about what these fish are doing. This is really important because bower construction is expressed over long time periods, and so you kind of have to follow these fish as they naturally express the behavior. It's unlike a lot of behaviors where you can introduce an animal and it will just perform the behavior right away, or you can inject it with something to trigger the behavior. So I'm going to tell you about the experiment that I've been working on to understand the neural basis of bower construction. And I have to say that I've worked with amazing graduate students, George Gruenhagen and Brianna Hegarty, who's not here right now, but none of this work could really happen without a whole effort from our team, and it's really fun working with them. So we monitor these fish downstairs in this building, and they're allowed to freely behave and interact with sand and females. And eventually, males will start performing this behavior, and when they do, as long as it's within a certain time window during the day, I go down there and I microdissect the building males, telencephalon, and also a neighboring male who is not building, telencephalon. And we run it through a single nuclei isolation protocol that we've optimized, and we sequence the RNA from the nucleus, not the cells, on the 10x platform. The behavioral data looks really good. When we go back and ask, after we collect the brains, we can analyze the 10-hour videos and the depth data and see that all of the building males that we collected were building, and we can see that in terms of both the predicted build events from action recognition in yellow here, and also from the depth change analysis of that's generated by our depth sensor. And before I go on, I just want to say that we also know that building males, a lot of behaviors in nature are complicated and co-vary with physiology and other behaviors, and that's the case with bioconstruction too. So these are relevant biological factors that we need to pay attention to if we really want to understand the neural basis of castle building. So building males are also performing more quivering behaviors, and they also tend to have a greater relative gonadal mass compared to control males. So when we get the data back from 10x, we need to make some sense of it. I'll kind of go fast through this because I know we don't have a lot of time. We clustered the data, so we use global transcriptional profiles, which separate our cells into clusters. We can also track canonical markers of core cell types that populate all neural tissue and see that these cell types nicely segregate across our clusters. We can also identify novel genes that are exclusively expressed in specific clusters. And this is my favorite way of understanding clusters. And this effort, this has been an amazing effort by Brianna Hegarty, who has gone really deep through hundreds of papers in Telios, identifying which genes out of a long curated list that we had show conserved expression patterns in cichlids and other Telios species in specific brain regions. So these are genes that we know about their molecular function, we know about their behavioral relevance in many cases, and we also know where they're expressed in the brain. They also exhibit biased expression patterns across our clusters. So we can get a really multidimensional biological understanding of what our clusters are. And then that is a nice foundation for starting to probe how do these clusters alter their function during behavior. So one of the classic tools in behavioral neuroscience for doing this is tracking IEG expression. And yeah, Michelle, you may notice that many of these genes are overlap with many of the genes that you just showed. So in neurons, depolarization triggers transcription of immediate early genes, and they can be used then in the brain to track which populations are selectively activated in response to a stimulus or during a certain context or during the performance of a specific behavior. And so I won't get into it, but these genes are actually kind of hard to track in neurons. And so I developed an approach to identify a module of these genes that strongly covary with one another across cells and across different clusters and cell populations. And in doing so, I think I've identified 7 genes whose immediate early gene-like function has not been described before. That's pretty exciting. So we're going to follow these genes in the brain in association with building to identify potential cell populations that are activated during building. And so we actually see one population, 9-GLUT, that shows a signature of activation. We use a pretty strict modeling approach, and we also account for variance that's explained by quivering and gonadal physiology. And we see that distinct subsets of cell populations are showing signatures of neuronal activation in association with these different biological factors. Another way of probing the behavior-associated function of our clusters is using unbiased differential gene expression analysis. And long story short, we again see distinct subsets of cell populations that show a disproportionately large number of differentially expressed genes in association with building, quivering, and gonadal physiology. And we also see that 9-glute, which was our only population at the clustering level that showed a signature of neuronal activation, also shows a disproportionately large number of differentially expressed genes. So multiple converging lines of evidence on that population and its importance for Bower construction. We can ask about what these genes that are differentially expressed in the brain in association with building and quivering and gonadal physiology are doing. We see many biological categories that are enriched, and many of the same categories are enriched among DEGs that are associated with building, quivering, and gonadal physiology. So it seems like similar biological processes are being recruited in distinct cell populations in association with gonadal physiology and with multiple different behaviors. So I don't have a lot of time. There's a lot of interesting things to talk about, but I want to talk about one follow-up that we're doing or that we've done on neurogenesis, which is one of our biggest hit categories for differentially expressed genes. And we have followed up on this in two ways. The first is, neurogenesis is really interesting because this is a mechanism that's been shown to be really important for behavior in mammals and fish across vertebrates, and it's also something that in humans slows down a lot as we age, and in adults is really limited in the brain. And so you can see there's actually many cell populations that are showing what you're looking at here is enrichment for behavior or gonadal-associated expression of 88 conserved genes with an annotated Go function of positively regulating neurogenesis across vertebrates. So same Go annotation in mice and zebrafish. And we see that a huge number of clusters show increased transcription of these genes in association with building, and a few clusters in association with gonadal physiology. And again, we see 9-glute, so which is the cell population that also showed signatures of neuronal activation and differential gene expression, or disproportionately large number of building-associated differentially expressed genes. So now 3 converging lines of evidence for that cell population and its importance for building behavior. So we also happen to know a lot about the process of neurogenesis in the brain, and we know that new neurons are being generated from radial glial cells in the telencephalon, and radial glial cells are localized to one cluster in our data. And we can ask about what that cluster looks like in behaving versus control males. And there's a classic signature of the transition of radial glial cells from quiescence to activation. So radial glial cells, they stay dormant and quiescent and then become activated and start generating progenitor cells, which can differentiate into many different types of neurons in the telencephalon. And this transition in fish is marked by transcription of aromatase, or CYP19A1. And we see a really strong and building-associated signature of activation of radial glial cells in association with building behavior. And we can even trace this signature down to specific subpopulations. And there's a lot of interesting things going on in radial glial cells that I can't get into, but the aromatase signal specifically traces to this subpopulation in green when we recluster them. And this is a subpopulation that is marked specifically by expression of the transcription factor LHX5, which in mammals, mice, plays an important role in hippocampal neurogenesis. So, in summary, I hope that I have convinced you that single-cell omics technologies, I think, are really disproportionately transforming neuroscience, especially because such a big problem for the field has been the heterogeneity of these cells that are all interspersed. It's very difficult, and no tool has ever allowed tracking the function of so many cell populations simultaneously in a single animal. And so I've shown you that specific populations exhibit transcriptional signatures of neuronal activation in association with building, and specific populations also exhibit signatures of neurogenesis, both neuronal and non-neuronal populations. And with that, I'd like to thank Dr. Han for organizing, Brianna and George, amazing teammates, Todd and Patrick McGrath for mentorship, funding agencies and the cores that have helped us along the way, and happy to take questions.
Thanks, Zach. Do we have any questions? Yeah, I'll start here.
Hey, Zach, very interesting talk. Thank you. I found your data related to radioglial and neurogenesis very interesting. I'm just wondering, is there evidence to suggest that neurogenesis, adult neurogenesis, is required for the castle building behavior?
There's no evidence of that yet. Honestly, there's not a whole lot known about the neuroscience of castle building behavior in general. It's almost like we're entering this system largely from scratch. And this is like our first pass blast at the brain and understanding the the neuronal functional basis of castle building. Yeah, so no manipulative experiments have been done, and there was a single other experiment using whole brain RNA-seq in a small number of animals has looked at Bower construction. Yeah, that's a good question. I think it would be a really cool whole line of follow-up studies that the data all kind of points towards neurogenesis if you just follow blindly.
Zach, congratulations on this. This has really moved along beautifully in the last few months. And I thought that the partitioning of the gonadal signaling and the quivering and the Bower was just really special. Mm-hmm. So two questions related to that. So one biological is, what are your thinking as to why there's a relationship between gonadal mass and neuronal expression? And second one is, methodologically, were you using MAST, or what were you doing to do that partitioning and separating it?
Yeah, I will. So the first question, what do I think is happening with gonadal? I assume maybe the neuronal activation signatures, for example. That's one of the most non-intuitive ones, I think. And what I think is happening is that I think that during building, their gonads tend to be larger, and they're releasing more testosterone, which is being converted into estradiol by aromatase in radioglial cells. Estradiol can permeate, since it's a steroid hormone, it can permeate the cell membrane and kind of go to outside cell populations, where it has been shown to prime neurons and increase their likelihood of firing. So I think that that's what the gonadal effect is likely. It's like a priming of many neuronal populations that may be important and involved in the circuitry of mating behaviors and other behaviors that are linked to the gonadal physiology changes. And then how am I doing it? Basically using a sequence of linear mixed effects models where I allow, it's not a lasso approach, but it's just a mixed model approach where I use like 6 different models and allow the variables to compete against one another in all possible pairwise combinations, and then I identify the effects that are significant in every single model. So the building effect is significant regardless of whether It's analyzed as a binary variable, building or not, or a continuous variable based on their level of activity. And when it's competing against gonadal physiology to explain variance, when it's competing against quivering, and when it's competing against both simultaneously.
All right, thank you. I think that's all the time we have for questions, and we can move on to the next speaker.
Thanks, George.
Hello, is this on? Okay, good. I'll just introduce Brett while he's getting set up. It's my pleasure to introduce Brett Aiello. He's a postdoctoral, NSF postdoctoral fellow in the Sponberg lab, and he's done a lot of great work studying the evolution of strategies of flight in moths and the evolution of wing shape. He got his Ph.D. at the University of Chicago in the Department of Organismal and Biology and Anatomy with Melina Hale. And today he'll be talking about the evolution of blinking in mudskippers and tetrapods.
Thank you, Ethan. I appreciate the kind introduction, and thank you, everybody, for being here. So today I'm going to talk to everybody about blinking. And I truly think blinking is one of the most underappreciated behaviors that a lot of us take for granted. And over the course of this talk, everybody in this room is going to blink on average once every 5 seconds, which is about 12 times a minute. But now that I mentioned that, some people in this room are probably blinking a lot more. Other people in this room are probably trying to hold back from blinking. If you're in that second group that are trying to hold back from blinking, stop doing that. Blinking is critical for maintaining eye health and epithelial cell function. And in many extant tetrapods, blinking serves multiple functions. One of these is eye wetting, or maintaining a film of fluid over the cornea, which is again important for epithelial cell health and oxygenation of those cells. And another is cleaning the eye of debris, which can be critical to maintain visual acuity and limit damage. And blinking can also function to protect the eye from foreign objects that could scratch or puncture the cornea. And finally, blinking invertebrates can serve as a way to communicate or interact socially with conspecifics, which is a behavior that we humans use pretty regularly and has even been discussed by Darwin in some of his writings. But since the origin of this novel multifunctional behavior, blinking and its associated morphology has diversified incredibly across tetrapods. For example, in humans, eye closure is accomplished by lowering the upper eyelid downward, where in some turtles, it's actually the lower eyelid that is raised to accomplish eye closure. And in many species of amphibians, reptiles, birds, and mammals, a third membrane, the translucent nictitating membrane, has evolved as a way to serve these exact same functions while allowing the maintenance of some visual acuity because of its translusiveness. However, hypotheses of how and why this behavior originated in early tetrapods are almost non-existent. We believe that blinking has only evolved once in tetrapods, and that the evolution of terrestriality in vertebrates is a strong selective pressure driving the evolution of this behavior. And we have several lines of evidence to support this hypothesis. First, to our knowledge, most fish do not exhibit a blinking behavior. Although blinking has evolved in some shark species, and it's believed to act as a way to protect the eye as sharks are attacking their prey. A second line of evidence is that in species of reptiles and amphibians that have secondarily evolved to be fully aquatic, they've re-entered the water full time, In all of these lineages, like the species of frog on the right, blinking has been repeatedly lost when these lineages reinvade the water full time. And the third piece of evidence is that the mudskippers are a second lineage of actinopterygian fish, this time the second lineage of fishes, this time within the actinopterygians, that have also evolved to spend the majority of their day on land. And similarly, mudskippers have convergently and independently evolved this same blinking behavior. So at this point, we know mudskipper blinking is phenomenologically analogous to blinking in tetrapods, but is this behavior actually convergent? So to support a hypothesis of convergence, we need to know how and why blinking has evolved in mudskippers, and for the remainder of the talk, we're going to do just that. So first, we're going to focus on the kinematics and movement of the eye, and then we're going to take a deeper dive into the associated morphology to see if any morphological adaptations have evolved in tandem with this behavior that could be facilitating these eye movements. Next, we'll focus on determining the functions of blinking in mudskippers, and based on what we know about blinking in humans, we're going to specifically test the hypotheses that in mudskippers, blinking functions to wet the eye and the corneal surface, clean the eye from foreign debris, and finally protect the eye from puncture by foreign objects. And before we continue, I want to recognize the amazing group of undergraduates that I've had the privilege to work with over the last 3.5 years. And these are the people that have conducted much of the work that you're about to see through their experience in the multi-semester vertically integrated project course here at Georgia Tech, where we're integrating these authentic research experiences directly into the curriculum. And this is this amazing innovative educational activity that I think really reduces the barriers for undergraduates seeking research experience at Georgia Tech. And I'm always so impressed by what comes out of these courses. So over the last 3.5 years through this project and others, over a quarter of the students that have participated in this class are now on published products, which I think is absolutely wicked cool. So let's dive into the how. The first thing we did was we examined the kinematics of mudskipper blinking, the motion of the eye and the associated apparatus that's responsible for blinking in the mudskipper. And as we can see by looking at the blue traces in this figure, mudskipper blinking is achieved by the ventral retraction or the depression of the eye downward into the orbit. And as the eye moves downward, a membrane below the eye, known as the nictitating membrane, is displaced laterally and dorsally as this behavior happens to completely cover the globe of the eye. And to investigate how this behavior is accomplished and whether any new musculature has evolved or any new glands or secretory cells have evolved in tandem, we conducted contrast-enhanced CT scanning of the soft tissue of the eye apparatus, and we find that mudskippers have not evolved any new musculature to accomplish this behavior, and they rely on some combination of their 6 extraocular eye muscles. Those 6 extraocular eye muscles that are well conserved across vertebrates and are the same muscles that you're using right now, for example, if you were rolling your eyes at me during this behavior, or during this presentation, rather. So next, we further analyzed the scan, and we determined that mudskippers have not evolved any tear glands that could be analogous to the lacrimal or meibomian glands of humans and other vertebrates. And we also confirmed this through histological staining and imaging of several areas around the eye and other parts of the body, and again found no evidence of glands or a high concentration of secretory cells around the eye. We did find the presence of mucus-secreting goblet cells, which are those highlighted on the bottom right of this histological section and indicated by the red arrows, but they aren't in any higher concentration here than they are at any other part of the body. So we find that in mudskippers, blinking is achieved through the ventral retraction of the eye into the orbit where it's passively covered by a ventrally located membrane. And this demonstrates that an incredibly complex behavior can evolve in systems with rudimentary morphology and suggests that few morphological changes or specializations are needed to occur for this behavior to actually evolve. So while we've gained insight into the mechanics of the blinking behavior and the associated morphology that facilitates this, we don't yet know the functional roles blinking plays in mudskippers and whether it serves some of the same purposes as it does in other tetrapod systems. So the next question that we wanted to ask was, does blinking function to wet the eye? So to do this, our team created an experimental chamber that was equipped with a computer fan at the top of the chamber and a small pool of water at the bottom of the tank. And mudskippers were left in the tank for a total of 2 hours. For one of the hours, the fans were off, and for one of the hours, the fans were turned on. And we calculated that turning on the fans increased the evaporation rate of a thin fluid film by approximately 20 times. And to ensure that the length of the experiment didn't impact our results, we alternated whether we turned the fans on for the first or the second half of these experiments. We then reviewed the film to identify the timestamps of each blink throughout the test period, and we calculated the inter-blink interval, which is the duration of time between 2 consecutive blinks, and then we compared that between our 2 conditions, fans on, or our high evaporation condition, and fans off, which we'll call our control condition. And we would predict that an increase in evaporation rate within the chamber should increase blink rate and therefore decrease the interblink interval. And that's exactly what we see. We found that the interblink interval is significantly shorter in high evaporation conditions. So mudskipper blink rate goes from about 1 blink every 2 minutes in control conditions to 2 blinks in a single minute under high evaporation conditions. So together, these data support the hypothesis that one function of mudskipper blinking is to wet the eye. The next thing we wanted to test was whether another function of blinking is to clean the eye. This is important because mudskippers, as you may have guessed from the name, they love the mud. And keeping the eye clean is critical for maintaining visual acuity for locating predators, prey, and conspecifics. So to test this hypothesis, we conducted a very simple experiment. We sprinkled as many brine shrimp eggs as we could over the corneal surface. We chose brine shrimp eggs because they're approximately 200 microns in diameter, which is within the size range of the sand and dirt particles found in waterfront environments, and is a completely harmless foreign particle for the mudskippers. And then we waited for the mudskipper to blink. And then when we reviewed the videos of these experiments to calculate the number of egg particles covering the corneal surface, both before and after a blink. And when we consider all of the blinks we recorded, we find that a single blink is highly effective at cleaning the corneal surface, and in most cases could completely clean the eye surface of all of its foreign debris. So we can now also support our second hypothesis that another function of blinking in mudskippers is to clean the eye. A third functional hypothesis that we wanted to test was that blinking can also serve to protect the eye from foreign objects that may actually act to puncture the eye. So to do this, we created a capacitance sensor, like one that you would find in the screen of a touch screen on your iPhone or some other type of phone or pad surface. And when that sensor touched a conductive surface, like the body of an animal or the eye of a mudskipper, it would illuminate a small LED, which we also kept in view. And we filmed ourselves using a high-speed camera lightly tapping the upper corneal surface of the eye, which would reliably trigger a blink every single tap. But we wanted to take this a step further. We wanted to determine if this behavior could actually be a reflex. So from these videos, we calculated 2 variables. The first variable was the lag time between when the eye was actually tapped and when the blink was initiated. And next was the duration, or the speed, of the eye closing period, which is the duration it takes the eye to be fully depressed during a blink cycle. And these metrics, lag time and depression duration, have been measured for the corneal reflexes in humans, which is the only other system we found data to compare this to. So we can now compare these data between mudskippers and humans to see if the temporal patterns of mechanically stimulated blinks in mudskippers are similar to those reflexive blinks in humans. And what we find is that the lag time averages just under 30 milliseconds in mudskippers. And this is very similar to the lag time of the corneal reflex in humans. We see a similar average and we see a similar degree of variability within the data. The next, we found that the duration of the depression phase, or the duration it takes to close the eye, is significantly shorter and therefore faster in mechanically induced blinks in comparison to spontaneous blinks. And these spontaneous blinks are the ones that we recorded for the kinematics that you saw at the previous part of this talk. And we found that, yeah, again, the duration of the depression phase, or the duration it takes to close the eye, is significantly shorter in these mechanically induced blinks. And in humans, we see a very similar trend. If we look on the y-axis on the plots on the left, the y-axis represents duration, and the left plot represents spontaneous, and the right plot represents reflexive blinks. And you can see that the duration of the eye closure period decreases slightly during reflexive compared to spontaneous blinks in humans. And velocity is also greater, which you can see in the second set of plots, the y-axis in the second set of plots representing velocity. And again, spontaneous is on the left, and reflexive is on the right. So combined with the high likelihood that the fully aquatic relatives of the mudskipper have not evolved any sort of eye retraction given their anatomy, we can now suggest that this represents the evolution of a novel reflex. So we can now also support our third hypothesis that another function of blinking in mudskippers is to protect the eye, which has likely occurred through the evolution of a novel reflex loop. And we could talk more about that later if anybody has any questions. So by studying this extraordinary behavior in a system where it is convergently and independently evolved, I think we could start to make some hypotheses of the different ways this behavior could have originated 365 million years ago when early tetrapods were making their first forays out of water and starting to invade the terrestrial ecosystem for the first time. The first thing we demonstrated is that a complex behavior can evolve in systems with rudimentary morphology. This suggests that few morphological changes or specializations were needed to occur for this behavior to actually evolve in early tetrapods, and that the origination of this behavior could have occurred through a simple eye retraction mechanism similar to what we see in mudskippers. And that's because most reconstructions of early tetrapods find that the orbit for the eye is located on the dorsal surface of the skull, which is often a really flat-shaped skull similar to what we think about in alligators. Next, it demonstrates that it's possible for multiple functions to evolve through the evolution of a single behavior. Multiple functions to evolve through the evolution of a single behavior. In mudskippers, we saw three totally distinct functions that are being performed with great efficiency by deploying the exact same behavior and by using the same associated morphology and neural control across all three of these functions. This suggests that it's possible for multiple functions to evolve as a suite rather than as a stepwise process, and that multiple independent selective pressures for different functions can simultaneously drive the evolution of a single multifunctional behavior. And because of that, that's why I think that this relatively complex behavior can evolve independently and repeatedly in lineages that are separated by many millions of years. So with that, I'd like to thank everybody in the Sponberg lab, the Shubin lab at the University of Chicago, the Baumel lab here at Georgia Tech Animal Care team. Thank you, Dr. Hahn, for the organization. And thank you all for being an incredible audience. I appreciate it. I'm happy to take any questions.
Can do one or two questions.
Yeah, really cool stuff. One thing I'm just wondering, in the beginning you talked about blinking evolving once or twice a small number of times, but if it's so easy for it to evolve, would you sort of question that? And the idea is that, what if you have species that go from an aquatic environment to terrestrial and then over millions of years sort of go back and forth? The idea is that if it seems from a physiological standpoint, it's pretty easy to evolve. And so does that make you think that maybe this is something where there's not just a single origin, but it could be many, many different times?
So great question. So across vertebrates, it has originated multiple times. We know that it's originated independently in mudskippers. We know that it's evolved at least 2 or 3 times in sharks. And in tetrapods, there has only been one transition from water to land. And therefore, we think that, along with all of the other evidence of it only being lost in extant species that have regained fully aquatic lifestyles, suggests that it has only evolved a single time in tetrapods. So all the tetrapods we see on land, we believe that that is just a single origin for that reason and those multiple lines of evidence that we went through. But it is this evolvable behavior, right? We do see it multiple times in these other aquatic systems, which really drives the point that blinking doesn't just function to wet the eye. it's also a very important function or behavior for protecting the eye and things like that. Yeah, but great question. Thank you. Oh, sorry.
Okay, thank you. So is there any variability in the depth of eye depression depending on the function of the blink? Because I think this is a really interesting difference between humans and a lot of other animals. Because they depress, it's almost like they depress and they blink. Whereas because there's a film covering it as well, so the depression is interesting.
Yeah, so you're asking, is there any variability in how far they depress that in during a blink? Yeah. So in our data for the spontaneous blinks, we saw a highly consistent depression amplitude, and we also saw that exact same depression amplitude in all of our mechanically stimulated blinks. So we know that at least in those two functions, it is about the same amount of depression distance. We didn't calculate the depression in the clean, but given the removal of all of the debris from the eye surface, it also suggests that it is a completely full blink all the way down. We think that it's actually a passive mechanism that raises the eye back up out of the skull, because in a cadaver, I'm actually able to depress the eye fully, and then it's able to spring back up. So it feels like it's almost this bistable system where you're either down or you're up, and it's very hard to lay yourself in between these 2 points. Great question, thank you.
Maybe we can do one more quick question, and that's it.
Going back to the evolution, I was just curious if there's anything known about, or if you would have any predictions about blinking in mammals that have transitioned back to water, like
yeah, great
mammals,
question. Like say what was the last one?
Dolphins, whales, hippos, or things like that.
Yeah, we don't have a lot of data on those particular species, those fully aquatic mammals, but mammals like beavers, for example, and other semi-aquatic mammals that are going in and out of terrestrial and aquatic environments, they've evolved that third nictitating membrane where it's this third membrane that goes like this across the eyes, and it's that translucent membrane, so they can continue to see. So when I think about these semi-aquatic mammals, I always tell people they've actually evolved like a set of goggles for themselves so that they're able to keep their eyes really clean when underwater, but maintain that visual acuity. But yeah, we don't actually know a lot about the dolphins and whales. It's an interesting group to look at. Yeah. Thank you, everybody.